So as we wait for the media center to recalibrate all of this, so it'll project, you guys will be flying blind unless you have a login, which since Mobina has a laptop, all of you guys will be clustered around her. But we'll go ahead and get started. So where we left off, we're talking about bulk transport, right? And so the nice thing about where we're starting on now is we've already actually covered bulk transport. We just didn't know it when we did it, right? So we already have talked about some of the elements of endocytosis and exocytosis. So the one thing to remember is bulk transport is the movement of large molecules, right? Up to this point with both passive and active, we've been moving very small molecules, right? Like ions and things like that. And so we've been able to like put holes in the cell membrane and be able to kind of get them through. But now we're talking about molecules that are a little bit too big, right? So if you were to do the same trick and put like a hole in the membrane to get some of these big macromolecules through there, it would leave, you have to put such a big hole in the membrane that would compromise the integrity of the cell. So you have to kind of come up with a different system in order to be able to pull this off. And that's kind of where we get to endocytosis and exocytosis. And basically all that really means is that in endocytosis, what you have is basically these macromolecules, these big bulk macromolecules coming into the cell itself. So literally you have basically a cell, and then all you have to do is kind of pull the membrane in a little bit. So you can imagine if I kind of pulled on a little bit, it would kind of pucker. And then if I continue to pull, these shoulders are going to meet, and then it's going to form like a little vesicle. Right. And so that would be endocytosis. Exocytosis, you guys have already seen. Right. So we're the secretory pathway. When the secretory cells were moving from the Golgi apparatus out to the periphery of the membrane, that was exocytosis. So you're basically spewing stuff out of the cell itself. And so both of these processes don't come for free, right? I mean, nothing in the cell does come for free. And so it requires energy, but it's kind of a different energy sort of profile than what we saw with active transport. That's the reason why we don't consider bulk transport as like true active transport, even though it requires energy. Because its energetics is kind of a little bit different. It's a little more indirect in nature. So when you take a look at the three different types of endocytosis, where you have phagocytosis, where literally you have the engulfment of like a bacterium, for instance, right? So as you're kind of sucking in that little bacterium, then what's going to happen is eventually you're going to kind of surround them, and that little bacterium isn't going to be completely surrounded. And then you can fuse your lysosomes to that and dump those hydrolytic enzymes. And you can chop up that bacteria. That's exactly what your white blood cells are doing. And if you can see kind of this image up here, I'll kind of blow it up. You can see this is an electron micrograph of exactly what's going on there, right? So it's an electron micrograph of a white blood cell that's engulfing a bacteria. Okay. Now, penocytosis is kind of the same idea. So this is phagocytosis. In penocytosis, you have kind of the same situation, only in this case, instead of taking in like a large particle, right? Like instead of taking in like a bacterium or something like that, what you're doing is you're kind of taking like little sips of your extracellular matrix. So a lot of times when you see a cell that's in the middle of penocytosis, you'll see not like one big endocytotic cell that's sort of sucking in something big, but what you'll see is a whole bunch of little ones like this. And it's basically just kind of sucking in just whatever happens to be out there in your extracellular matrix. And so this is a really, really good way for the cell to kind of sample what's out there. Because remember, the cell is the one who actually made the extracellular matrix, and it has to maintain it, right? So if your needs change, and usually laying down your extracellular matrix is a dynamic process, so you don't just lay it out there and forget about it, right? You're constantly adding to it, you're taking away from it, you're mixing it up a little bit. And so in order to know exactly what you want to put out there, you have to kind of know what you have out there already, right? And that's kind of what pinocytosis does. So it kind of allows you to be able to sort of suck in a little bits of this extracellular matrix so you can kind of get an idea what you have out there. It also just randomly brings in whatever cellular molecules are out there. So if you just need doesn't really matter. You just need some amino acids, maybe some proteins floating around out there. You just suck them in and you're good to go, right? So that'll be like a little fresh reservoir of amino acids. And who doesn't like a good amino acid shake, right? That kind of thing. And so this is over here, a nice example. Now these are colorized, right? So these electron micrographs, they're not actually that color. They've colorized them so that you can see what's what. But in this particular case, you can see these smaller vesicles that are pinocytotic in nature. So you can kind of see the stuff that's going to get pulled in. So that's another one. Now, the one that kind of threw you guys for a little bit of a loop, because it's one that we haven't really seen a lot before, is the receptor-mediated version of endocytosis. And this is kind of like the same idea, right? So that there's the common denominator for all of these is you're forming a vesicle, you're internalizing something in this particular case. Oh, yes, thank you very much. Thanks for working on that. So in this particular case, what you're going to do is instead of just kind of randomly taking something in, whichever happens to be out there, you're kind of like specifically wanting something in very, very specific need for it. So you're not just looking at random molecules that are out there, you're looking at one specific molecule out there. Well, if you want to reach out and grab that one specific molecule, you got to make a receptor for it, right? Which is kind of like, basically a protein hand, if you will, for the cell that can kind of reach out and specifically bind to that particular molecule. And that's kind of what you're doing. So here you can see your receptors that are basically out here on the cell surface, and you're going for this one particular target molecule. You're blind to all the rest of the molecules that are floating around out there. So like, for instance, there may be other molecules that look something like this. It doesn't really matter because your receptors aren't going to touch those. Right, or maybe there's a couple of them out here that are like this, little square-like things. It doesn't matter because your receptors aren't going to touch those. The receptors are going to basically sift through the pile, and it's only going to grab the ones that it's designed to grab onto. Now, once you actually grab onto your molecule, which you can see you progress to the next stage of vesicle formation here, then it's time to internalize it. Right, but here's the problem. What happens if I just grab onto the receptor and I just yank on it? Right, that's one way to internalize it. Well, the problem with that is what's going to happen is it's going to tear through the membrane. So you're not going to really achieve anything other than just damaging your membrane, and you'll get one receptor with one molecule on it, and that's not very effective. So what you do instead is you embed your receptors into a foundation of a protein called clathrin. So this is kind of like a structural protein. It's kind of like taking a mailbox, right, and sort of embedding it with a cement footing. And that way it doesn't tip over, fall over, or do damage to the lawn or anything like that. It's kind of the same situation. Only now what this does is this clathrin foundation gives you something to grab onto so you don't damage the membrane. So now what happens is instead of grabbing onto the individual receptors, you grab onto the clathrin coat and you pull on it. And so basically that'll allow you to disperse your forces throughout the clathrin foundation so that you're not pulling that membrane apart and shredding it. So once this internalizes, then what you get are these little clathrin coated pits. With your molecule of choice inside. And so here you can see the clathrin they've highlighted in blue here. So you can see this little clathrin coat around this particular pit. And you can see these vesicles that are in various stages of development up here on the membrane. So that's kind of good, right? Because you got two different mechanisms to just bring in stuff, big stuff or smaller stuff. And you've got one where you can actually be very specific about what you bring in. So between the three of these, you actually have a lot of control and regulation over what's coming into the cell. Now, exocytosis is easy because it's just basically the reverse order, right? So for instance, in this particular case, remember we had the secretory pathway that we finished off with in the last chapter. And at the end of the secretory pathway, we had our secretory vesicle, which merged with the plasma membrane, right? Like two soap bubbles merging together. What happens there is, of course, they unite, right? And they merge together. And then when they merge together, it basically turns your vesicle inside out and it spews all of your contents to the outside of the cell, right? So that would be an example of secretion, which is usually by exocytosis. And this is probably one of the coolest electron micrographs actually we have in the book, because this is actually an exocytotic vesicle caught in the act of secretion. So you can actually see, and it's very hard to do this, by the way, because remember, when you're doing electron micrograph sectionings, you're just sort of like randomly taking sections and you just hope you get something good. And so there's this getting something like this is like a 1 in a million chance, but you happen to have caught it at the exact moment that it is basically ejecting its contents to the outside of the cell. And you can also see how the membrane of the vesicle has now fused with the membrane of the cell itself. So you can see that kind of it's turning it inside out kind of an effect. And so this is like a really, really great image because it kind of shows you what's going on and how it actually looks. So when they draw this little cartoon over here, it's not just like artist's interpretation. It actually looks like that when you catch one in the middle of exocytosis and secretion. So you can already see it. Hopefully you're kind of gaining a little bit more of an appreciation for just how powerful electron microscopes have been for us in understanding cells and cell structure. We didn't have any of that before electron microscopes, right? So every single one of our organelles in the last chapter had electron microscopes, right? Images for us to see that. So this is like for you table people, right? For those of you who like tables and you like to use tables as your organizer, this is it, right? So you got your passive, your active processes, and then of course your bulk, all kind of lined up in a nice little table. Okay. So that was easy. That was fairly painless, wasn't it? Okay, maybe not. Oh, now it starts to get good. Now the semester begins. Of course, that's because I'm a joint person. See, thank you. So I'm not crazy, at least not for that reason. Right? I'm crazy just for other purposes and other reasons. Okay, so this is basically chapter 10. So a lot of times you'd be like, well, wait a minute, what happened to 6, 7, and 8? Well, 6, 7, 8, if you recall, are the energetic chapters, and that is part of our sort of little side situation that's going on with our take-home exams and things of that nature. Okay. So this is an important chapter because the next 3 chapters are actually kind of a trio. They're kind of like like the Three Stooges, if you will, or maybe not so Stooge-like, but they're a trio. They go together, and it's helpful to understand them as a triad that goes together. So because all semester long, we've sort of talked off and on how the ultimate goal for any biological organism is to do what? To reproduce, right? But here we are, well into the semester, getting ready to wrap it up, and we haven't even talked about reproduction. Well, now we do, right? This is the first of those reproduction chapters, not for us, but for single cells. So if you're a single cell, for you, cell division is reproduction. Now, we're not single cell, right? So we have a little bit more work to do before we get to our reproduction piece. That's the reason why we have to take all 3 chapters together. So what are the 3 chapters? Chapter 10 is mitosis and cell division, right? You have to start with that one. Chapter 11 is meiosis, also part of cell division, but a slightly different cell division with very different goals associated with it. And it's usually wrapped into sexual reproduction, which is why meiosis is important for us, because we are sexually reproducing organisms. And then the crown jewel of, I would argue, all of biology and you already know my bias, and I claim it, and I don't back away from it, is genetics, right? Because ultimately genetics is the passing on of traits from one generation to the next. Now, what is the mechanism of how you pass those on? Well, actually, as it turned out, the mechanism of genetics is meiosis, which is chapter 11. So in order to understand genetics well, you have to understand meiosis well, because meiosis exists to serve genetics. Now, what if you want to understand meiosis well? Well, in order to understand meiosis well, you want to understand mitosis well, right? So your beginning knowledge starts now. So if you want to understand and not have any problems with meiosis, the best thing you can do for yourself is understand mitosis very, very well. You're going to make meiosis much easier. So let's take a look and see what we're talking about here. And we're going to be multicellular centric, right? So we're going to be looking at our cells because that's what interests us. So basically, when you take a look, and what I want to do is I want to sort of show you how the next two chapters, mitosis, meiosis, and genetics, kind of all sort of interwrap in and kind of wrap themselves around our entire sexual life cycle as multicellular sexual reproducing organisms. Because we all start off in the same place, don't we? We all start off here as a single cell. And how many chromosomes do we have? Well, you guys should know this now, right? Because you did those karyotypes, you did those chromosome squashes. So you got a chromosome from mom and from dad. So that means you have how many? 46, right? So you have 22 autosomes, which are what we call the non-sex chromosomes, right? So that's 22 times 2 is 44. And then you've got two sex chromosomes, either XX if you're a female or XY if you're a male. So that gives you number 45 and 46, right? So when you first start off life, here you are with 46 chromosomes. And the first thing you do, and for a good while, what you do is you essentially go through cell division. And you create two identical daughter cells. But guess what? You're not done yet, are you? Because you got a lot more than just two cells in your body, don't you? Yeah. So guess what? These guys also go through a round of cell division. Yes. And they do the same thing. They create two identical daughter cells, each with how many chromosomes? 46. And these will go through a round of cell division. And these will go through a round of cell division. And so on and so forth. Until eventually you're born. That's my little baby, right? That's like a little diaper that's kind of wrapped around there. They don't come out with diapers, by the way, just letting you know that. That's you're like, oh, that's great. Right. So right. So that's basically our body. And so this whole process then, where you just go through iterative processes of creating more identical cells, this is all mitosis. So mitosis basically builds the body. That's what it does. Now, in our body, there's a subset of these cells that are set aside, special from all the rest. And what these will do is these will be designated to go through the next type of cell division, which is meiosis. And there's two phases to meiosis, not just one. There's a meiosis 1 and a meiosis 2. But here what's going to happen is you're going to create now instead 4 non-identical daughter cells. With how many chromosomes? 23. So you can see it's already a little bit different, isn't it, than mitosis. So what do these 23 chromosome cells become? These become your sex cells. So if you're female, these become your eggs. If you're a male, these become your sperm. So if you take then the 23 sex cell from one individual and you pair it with the 23 from another individual, then these two will fuse together and then they will form a cell with 46. And guess what? You start it all over again. Only now, instead of you going through the bodybuilding, it's your kids going through the bodybuilding. And not only that, but the 23 chromosomes that you chose to pass forward has a certain number of traits, but not all of them associated with them. Same thing with your partner. They pass forward half of their traits, but not all of them. And they're randomized traits. And then when those two come together, then the 46 of the next generation has a different combination of traits than what you had when you started off life as a single cell. So that passing on traits from one generation to the next then becomes genetics, which is driven by meiosis. So you can see how all of it taken together is kind of like this giant sort of life cycle for us as an organism. Mitosis to build our body, meiosis to create our sex cells, and then genetics to have a mechanism to pass our traits on to the next generation. And it's only once you've successfully passed your traits on to the next generation that you can claim to have successfully achieved your task as a biological organism. You've successfully reproduced at that point. Okay. So that's kind of how they all tie together. So let's go ahead and take a look then at mitosis. So for us, for multicellular organisms, cell division is important, right? But it's important for different reasons. So for instance, obviously for us, reproduction and cell division are the same. For single-celled organisms. But for us, these are the multicellular ones. What do we do cell division for? To grow, right? Grow is basically adding new cells. That's basically what growth is. You're adding new cells. Where do those cells come from? Well, they have to come from cell division. Wound healing. In this case, you damage cells that need to be replaced. And of course, cell replacement. This is just basic normal turnover, right? So your cells, you don't have the same cells in your body that you were born with, right? I mean, a lot of your tissues get turned over. They probably, by the time you're an adult, you probably have turned over your cells a few times. Okay, so that normal replacement is going to be associated with cell division. So you constantly have to have cell division at work all the time. And so what we're going to do first is we're going to study what cell division looks like in prokaryotes and bacteria in particular. And then we're going to apply some of those lessons that we learned to eukaryotes to see if we can understand what's going on with cell division. So first of all, the important thing to understand with bacteria is that they don't have a sexual life cycle. They're asexual. And they're clonal. So they're literally just like mitosis is for us. So you'll take a bacterium, and then it will basically divide into two identical daughter cells, in this case, two identical bacteria, two clones of each other. So there's a couple of things, however, that you need to make sure that you get straight. So there's a mantra, there's an order of operations associated with cell division that you cannot violate. Because here's what you're doing. Notice what we did in cell division. We went from one 46 chromosome cell to two 46 chromosome cells. right? In order to maintain that chromosome number, what you have to do is you have to double everything and then separate it into two piles. That's the only way you can do it. Okay, so you got to make a copy of it. So that means that before cell division, which is simply separating the two piles, you always have to first start with duplication. So when you're talking about cell division, it's always duplication or replication first, then separation. So make twice as much of everything and then separate that twice as much of everything into two separate piles. That's how you get two identical piles or two identical cells. Now with the bacterium, you have to do the same thing, right? So for you, the first thing you have to do obviously is you have to duplicate your DNA. So that's a thing we have to talk about. And then we also have to duplicate the cell itself. We'll talk about that here in just a little bit. But let's take a look at the duplication of DNA first. In bacterial cells, their DNA genome is actually a circular type of a genome. So this is not, I'm not talking about the plasmid. This is like your main genome that's kind of all wrapped up in the nucleoid region. It's actually a giant circle that's kind of folded up on itself and just sort of wadded up. But it is a circle, which is helpful, right? Because if your first task is to duplicate everything, then you have to sort of, you sit down, you get ready to do this for the DNA, right? You get ready to duplicate or replicate your DNA. And there's a natural first question that should come to your mind. This is nice, it's a circle, but where do I start, right? I mean, is there a place to start here? Do I just pick some place and go? So is there a start point? And as it turns out, when we study replication in bacteria, what we realized is that there is an actual start point. It's called an origin of replication. It's a definable point or place in the DNA that will basically tell the replication proteins and enzymes to start here. It's like a big flag, start here for replication. Now, bacteria also have something else. They've also got a site of termination, so it's a stop site. We have both. We don't really have a stop site because we have slightly different structured chromosomes, but bacteria do have a stop site. And then basically what they do is they start on the origin and they just start making DNA until they finish the circle. And then once they finish their two genomes and they create a septum, which is like a wall, this is like a protein wall that divides the cell. So this is what it looks like. So here you can see you've got your origin of replication in green. So your replication enzymes will bind to that origin. And one group of enzymes will go clockwise and the other will go counterclockwise. And then what they'll do is they'll meet at the termination site, which is on the opposite side. And so here you can see there's both these guys going sort of clockwise and counterclockwise. And then basically they met here at the termination site. This is what I call the magician's ring phase, where they're kind of like linked together. And now all I like to do is basically separate these two apart from each other. And now you kind of get this cell with these two identical copies of DNA in it. So you successfully duplicated your DNA. We're going to talk more about the nitty gritty of replication in chapter 14. That's got its own chapter, so we don't want to talk too much about it here. But then what's going to happen is in order to separate the cell, you have to elongate the cell. And then eventually what you want to do is you want to pinch it apart right here in order to create two new cells. That pinch apart is what's created by the septum. So the septum you'll see is right here in the middle. There's just like this little protein wall that's made of a protein called FITC. It's actually not a wall, it's actually a ring. So if you turn it on its side, you actually see this like little ring. It's like a little cinch cord. And so what it does is it just cinches down on the cell until eventually it just pinches it apart. And this is an actual image of the septum. So you can see the This is going to be the Fitzy wall, but when they turn it slightly angular, you can see that it's actually a ring. And what happens is it's a contractile ring. And what'll happen is it'll just start to cinch down. And as it cinches down, it's going to take the membrane with it. It's going to pinch it apart into two cells. And then you have your DNA in the side, your DNA in the side. And then you just basically pinch the two cells apart. And you've created two separate cells. As it turns out, this Fitzy is actually something we see in most prokaryotes. So this seems to be like a prokaryotic thing. However, what about eukaryotes? So Fitzy actually is not in eucs. So how do they do this? Well, as it turns out, eukaryotes don't have Fitzy, but they do have a different protein. They have tubulin. And as it turns out, the structure of Fitzy is very similar to tubulin. So Fitzy is actually tubulin-like. So essentially eucs use tubulin instead. Also remember from chapter 4, remember we said the cytoskeleton, which is the section that tubulin is found in, is one of the big innovations of the eukaryote. And so here you can see how the eukaryote basically took sort of an idea from the prokaryotes and they kind of made it their own and they turned it into something completely different. So that's basically how eukaryotes do it. So instead of using Fitzy, they have a contractile ring, by the way. They have a way to sort of pinch the cells apart, just like the bacteria do, only in this case, they're using the tubulin instead. And microtubules associated with that tubulin to do this job instead of Fitzy. Okay, so let's take a look at a couple of different types of organisms, and let's talk a little bit about how they structure this mitosis and how they kind of pull this off, right? So we already talked about bacteria. We talked about how they do things, right? This is what's called binary fission. But remember, the eukaryotes have to do something different. Why? Because the one thing that eukaryotes have that complicates matters is they've got a nucleus, right? And you've got this nuclear membrane that's designed to protect your DNA. And so different organisms will have to figure out how to move their chromosomes around and how to deal with this protective barrier, right? This nuclear envelope, because obviously it's protecting your DNA, but now you also need access to your DNA. So how do you get access to your DNA and still keep it protected, right? That's kind of your problem that you're trying to solve. And different protists will do it differently. So basically, if you take a look at a plant-like protist, what they do is they basically organize their microtubules, notice, without a centrosome, right? Because plants don't have centrosomes. That's the reason why they're plant-like, right? But you have some sort of a microtubule organizing center. And basically, you organize a couple of microtubule types. So one is like what's called a thoroughfare microtubule that goes all the way through, kind of punctures, if you will, the nuclear envelope and sort of traverses the center portion of the nucleus. And then, of course, you have some flanking lateral microtubules that are designed to grab onto the chromosomes and pull them. Kind of like you're sort of poking through the envelope itself, right? So you're kind of reaching through the envelope and you're kind of grabbing onto a chromosome and you're kind of pulling it to either side. So that's the way these plant-like protists do it. The animal-like ones are a little bit different, right? Because now they've got centrioles. So they form their spindle with the centrioles. Same idea though. Notice they've got the thoroughfare microtubules, and then they actually puncture directly into the nuclear envelope and grab onto those chromosomes and pull them to the side. But notice in both cases, your nuclear envelope remains intact. Yeast, similar idea, slightly different location. So they build a little protein pad called a spindle pole body on the inside of the nuclear envelope, and it's from there that they build their microtubules that will grab onto the chromosomes and pull them, right? But notice they have the same pattern. They use kind of thoroughfare microtubules that go all the way through, and they build microtubules that grab onto the chromosomes to pull them. So there's two different types of microtubules that all of these guys are building. The thoroughfare ones that are connecting the two poles together, so the two poles are connected through the thoroughfare ones, and then the chromosome attached ones, which are grabbing onto the chromosomes and pulling the chromosomes toward them. In all cases, though, the nuclear envelope stays intact. Why would that be important? If you're a plant like Protus, like a euglena, for instance, or if you're an animal like Protus, like Paramecium or amoeba, or if you're a yeast and you're just kind of out there hanging out, different kingdom, right? Kingdom fungi, but same basic problem, right? Why would it be important for you to keep that nuclear envelope intact? Why not just destroy it and get access to your DNA and deal with it and get it over with? I have no idea. I'm not a protist. Think about it. Your most precious resource as an organism is what? Like if the house were on fire and you could grab one thing as a cell, what would it be? Your DNA, right? Everything else doesn't matter. You're going to grab the DNA. Why? Because if you don't have your DNA, you're dead. You literally don't have any clue how to conduct your biological business. Right. So this is like the most valuable thing that any cell or any organism has. So if you're a protist, what is life like for you? I mean, here you are, a euglena, swimming around in this pond out there, or a paramecium or an amoeba. Is life nice and cushy and peaceful for you? No. What's happening? Any good stuff happening to you? Oh, we're so sheltered. Life is so good to us, and we have no clue how good it is. No, everything's trying to kill you, right? You are like the bottom of the bottom of the bottom. I mean, so there are all sorts of things that are trying to eat you. There's, all sorts of things that are trying to kill you. That's before you get to the whole problem with the environment, right? I mean, you're swimming around in a pond, and I don't know about you, but it's kind of a little dodgy right now swimming around in a pond, because you never know from day to day whether or not it's going to be liquid or ice, right? Because or like even in the same day, right? So it starts off as ice and it turns into liquid. It goes back to ice again just within a few short hours. That's just the way spring is in Denver. Is that a pretty, that's a pretty harsh environment, right? But guess what? Throughout all of that, you basically have to make sure that the one thing you protect is what? Your DNA, right? Because if you lose that one, game over. Absolutely game over. So if you go through cell division, which by the way is a good thing, right? Because if you're a protist, cell division for you is reproduction. So that's a good thing. But guess what's not a good thing? Is if you leave your DNA exposed and something comes in and takes advantage of that, right? Aha, caught you with your DNA pants down. Now I'm going to get you, right? So that's a state of vulnerability that you cannot accept. So what do? You keep your protection up and you figure out how to deal with it. Okay. Life is not good to these cells. And then comes us. What do? We break down the nuclear envelope. We do the most efficient thing, right? Get the envelope out of the way. Just go grab the chromosomes, separate them, and you're good. How come we get away with that?
How come we can get away with this and these guys can't? Well, think about it. I could tell you why. I mean, what we just went over was a good illustration of why. I mean, you guys were having a hard time trying to figure out how bad life was for a protist because life is so good for you. Right? For your cells in particular, they got it pretty easy. Does your average cell have to worry about being attacked? No, it's all well protected, right? So all of your cells are inside your body. They're all protected from the environment, right? They're protected from bad guys trying to kill them. So for you, it's like nice and cushy. So yeah, you can drop your DNA pants and walk around a little bit. It's like a nude beach in France, right? I mean, there's no problem there. Nobody's going to arrest you. You try that here, you'll get arrested. I'm just saying, don't try that here, right? I mean, so it's just kind of a little bit different. So you're protected there because it's a thing, I guess, in France. I know this just not by experience, by the way. I've heard about it. That's about as good as the closest I've gotten to that one. But I just find beaches awkward places anyway. I can't imagine what it'd be like to be on a nude one. I mean, because it's like, yeah, because like half the time you're trying to like hide your reaction to like what people are looking like, and sometimes that's like, whoa, and sometimes that's like, you know, it's like a look of horror on your face. It's like, wow, I can't unsee what I just saw right there, and that's hard sometimes to sort of mask that a little bit. So yeah, find myself staring at the sand a lot, right? So yeah, just focus on the sandcastle. Yeah, but anyway, so that's the reason why, because our cells are internal, and so we can afford that because they're extra protected. Okay, and that's helpful for us. So then when we actually go to working with these chromosomes, this is a great electron micrograph of chromosomes that are actually in the middle of cell division. So they kind of look like cheese puffs, right? These are the cheetosomes that I always talk about, right? They kind of look like little cheese puffs, but they've already gone through duplication. These are the ones that are getting separated, right? And of course, they're not red and blue. They're not colored at all. They just color them red and blue for some reason to highlight, I guess, the maternal versus the paternal chromosomes. That's what they did with these ones. But we already know that for us, now each species has a different number of chromosomes, like fruit flies, for instance, which is a common genetic organism that we use in the lab to study genetics, has 4 chromosomes. We have 46, 23 pairs, right? So we have 22 autosomes times 2, which gives us 44, right? One from mom, one from dad. And then we have 2 sex chromosomes, which is either XX or XY. And that gives us 46. Okay. So that's our complement. And in some organisms, whether or not you're missing some can have various effects. Right? And as a matter of fact, we have an entire chapter in the genetics course that's running in the fall, where we go into this in a lot more detail. Typically in humans, having extra chromosomes or missing a chromosome is not tolerable. That it's catastrophic, actually. And if you guys remember your chromosome squash, and you had like a lot of different options there, like you had Patel, which is extra 13, and Edwards, which is extra 18, and Downs, which is extra 21. And then you had like XXY, and then you had XO, right? So as it turns out, with the autosomes, these are very intolerant to extras. Matter of fact, when you take a look at Edwards and Patel and Downs, of all of those extra chromosomal phenotypes, basically the way they look, the only one that's really viable is Downs. So most of the time, if you've got an extra chromosome or if you're missing a chromosome, it is a catastrophic fail in development. And normally those will lead to spontaneous abortion, which is this fancy word or the technical term for a miscarriage. Most of those will lead to a miscarriage in the first trimester. As a matter of fact, the overwhelming majority, the higher percentage of miscarriages in the first trimester are because of chromosomal abnormalities. So you can't even get to first base, much less past first base, at least in us. But for plants, it's a whole different ballgame. So plants can absorb and they could take on like lots of different extra chromosomes. As a matter of fact, most of the produce we buy in the store are not normal, like to wild type. Strawberries, for instance. I don't know if you've ever seen a wild strawberry that has 2 chromosomes from, one from each parent. But a wild strawberry is a little itty bitty thing. I don't know about you, the last time we bought some strawberries, got some in the refrigerator home actually right now. Those things are huge. You know why? Because what we did is we bred them to have extra chromosomes. So you're actually buying produce that has extra chromosomes in it. Which is kind of funny. It's always the whole GMO argument always kind of is a little comical to me. Because I look around, I'm like, you do realize that everything you're eating right now is all GMO in one way or another. We have modified everything that we consume. There is no such thing as a non-modified thing that we eat. Everything's been modified. Nothing is wild. So it just doesn't exist. Even if you go to and get seed from a farmer, those of the crops, those have all been modified and bred multiple times, and none of it's native. So that's just, I mean to burst your GMO bubble, but sometimes the GMO argument just kind of gets a little silly sometimes to me, because everything is largely modified. And I never really did quite understand how you pick and choose which ones to care about and which ones not to care about. So until that becomes clearer, then I just sort of well, whatever, I'm still going to buy strawberries and eat them. So, because they taste yummy. And if it shaves off a few years off my life, it's been good. A good run. Okay, so let's take a look at chromosomes now, shall we? So we already talked a little bit about chromatin, didn't we? We talked a little bit about how DNA isn't just a naked molecule. It's not just the twisted rope ladder duplex that we associate with the DNA molecule. That in reality, that it's wrapped around these little protein drum-like structures made of a protein called histones. And there's a series of histones that make up this little complex. But then what we do is we wrap our DNA around these little drums. And then each one of these units is what's called a nucleosome. So it's this little protein complex with the DNA wrapped around it. Now what this does is it gives our DNA structure so it doesn't get torn up as we're using it. Because remember, the DNA isn't just some stale, stodgy, dust-covered book in a library that nobody ever reads. This is a working document, right? I mean, this is actively being accessed. Everything you need to do in the cell, you have to go to the DNA in order to get the instructions to do it. So this is something that's heavily used. And so in order to minimize the wear and tear and the damage associated with it, you have to sort of protect it a little bit. And that's what this chromatin structure does. It basically kind of wraps this DNA around these protein protective drums, and it sort of protects them a little bit. Now, when you take a look at the chromosomes that we actually have, this kind of structure can have a couple of different looks, right? So you could imagine that when you have this sort of oftentimes we refer to it as beads on a string, but you can imagine that you could actually wind this up tighter and tighter and tighter and sort of condense it down, compact it even much more so. That would be very protective of the DNA, but it's not going to be very useful for you to use, right? Because you're gaining in structure, but you're losing in accessibility. And so when you do that, it refers to a type of chromatin called heterochromatin. So this is basically a tightly condensed chromatin. You've basically wadded up, but the genes in it are not accessible. And because they're not accessible, that means they're not expressed. That means you can't make a protein from them because you just can't get access to them. So it's what we call transcriptionally dark. It's an area that the genes just aren't active. They're silenced because of just the structure of the way you've wadded it up and you've made them inaccessible. Now, on the flip side of that, you've got what's called euchromatin. So this is a less tightly condensed chromatin with a looser, more accessible structure. So any genes in here are going to be expressed because you can get access to them. And so there's been a lot of research on what we refer to as chromatin remodeling. That is just basically, how do you go about the process of bunching DNA up into a tighter structure versus loosening it up, right? And what we noticed is that one of the ways you can regulate genes, either turn them off or turn them on, is sometimes by just remodeling the chromatin. So do you want to turn the gene off? Then just remodel the chromatin so it's more condensed and that gene's inaccessible and you've effectively silenced the gene. You want to turn that gene back on? Loosen up the structure, make the gene accessible, and you turn the gene back on. So we're starting to see that there's a lot more than just physical structure associated with this, that it's actually leading into a lot of control of how genes act and behave. Okay, there's a lot going on in there. So the nucleosome then is this sort of little drum piece with the histone drum with the DNA coiled around it. So you have 8 histone proteins that make up this complex, and you have about 200 nucleotides roughly of DNA wrapped around it. And the way that you get the DNA to wrap around it is by positive negative charge attraction, right? So the histones are positively charged. DNA, because of the phosphates, is negatively charged. So you basically get an electrostatic attraction. Why is that helpful? Why not just make it like a covalent linkage? Think about it. Do you need access to your DNA? Yeah, you need to basically take your DNA, you need to be able to open it up and read the content so you know which amino acids to pick for the protein you're trying to make, right? Sometimes you need that because you're actively making that particular protein. Sometimes you don't if you're not actively making it. So what you need is this DNA to be reversibly bound to this. So when it's not being used, it needs to have a nice connection. That's the electrostatic charge, right? So it is a nice connection. Remember the balloon stuck to your head, right? But at the same time, it needs to be weak enough so that you can pull it apart and read the code, the DNA code that will tell you which amino acids to pick. So you need a combination of both, right? You need structure to hold it together, but you also need it to be reversible and weak so that you can actually get access to it. And that's the reason why you use this electrostatic attraction. It provides structure and access. So this is kind of what it looks like, right? So basically this is your basic DNA molecule. This is your twisted rope ladder. Now, if you were to yank on this, it would shred, right? It'd be like pulling on a cobweb. You pull on a cobweb, what's going to happen to it? Just disintegrates, right? It's just like disappears on you. And that's what would happen with DNA if you did that. That'd be a bad idea because if you damage your DNA, that's going to create mutations. You create mutations, nothing good happens after that, right? So what you do then is you bundle it and you wrap it around your histone core. So that's your big core there. And you kind of pin it right there with another type of a histone to kind of hold those coils of DNA together. But remember, you can remove all of this. You can make it a little looser. You can make it a little tighter. It's a very dynamic type of a process. And we're learning that in research, how dynamic it is. It's quite a significant process. But when we're not dividing, what does your DNA look like? Well, remember, we already know that it basically has 2 different forms. It's either in a heterochromatic form, that's tightly condensed and gene expression is pretty much off at that point, right? Because of structure and condensation and accessibility to the genes. Or it's in a euchromatic state where it's a little more open and accessible, so you get gene expression out of that one. Well, when you have an actual, the nucleosomes themselves, when you're kind of organizing these in chromosomes, you create what's called a 30 nanometer fiber. And basically what this is a coil of chromatin that has a diameter of 30 nanometers. And so this is a little bit more structural, right? But you can loosen this up as well. If you need access to a gene, you can tuck it back into the 30 nanometer fiber if you don't need access to the gene. So this is all a very dynamic system if you can get access to all of this. But this is kind of your starting point, right? So this is kind of what it looks like in a non-dividing, just a cell doing its job, right? So it's just doing its thing. And this is where we start. But when we want to get dividing, it's a little bit different, right? There's a particular activity we want to do. We want to be able to move these chromosomes from one place to another, from one end of the cell to the next. Think about it this way. Moving a chromosome during cell division is a lot like moving a fine china set across country. So imagine that you're like an East Coast family that's been on the East Coast for generations. And you got this fine china collection that's like generations in the making. And you got like multiple generations of attitude. It's a significant china collection, and it's like a prized possession of the family. If you're going to move that to LA cross country, how do you move that china collection? You need to pack it very well, right? So you don't just grab it and start slinging, right? Because it's like precious. It's like they're antiques. It's like they don't make them anymore. So how do you pack it? Well, first of all, you're going to take like all the plates, right? And you're going to pack them together. You're probably going to put some packing between the plates so there's no plate on plate clinking, right? And then you're going to put all the plates in one area. You're going to fill in all the holes with probably packing popcorn and other stuff so that nothing in there is moving. You're going to do the same thing for the saucers and the other things, right, that you're going to have. And you're going to wrap that box up and you're going to put what? Fragile on it, right? And you'll probably do a couple of death threats, right? Break this and I will. And every generation after you that's related to you. It's a little extreme. But chances are you're not just giving that to the movers, are you? If it's something's that precious, who's moving that one? Yeah, that's coming with you, right? You're like, that box right there, don't touch it. That one's mine. I want this one under my wing the entire way to LA. And I don't want it to leave my wing. I want to make sure that this thing makes it to LA. Now, why you would want to move to LA, I don't know. But because I was a Northern California kid, I can't stand Southern California. But you have to do the same thing with your chromosomes, right? And then not only that, so what you're doing is you're taking your china collection, which is spread out and like on display. And then what you have to do is you have to condense it down to a couple of well-packaged, well-protected boxes. And then you move it, right? And then once you get to LA, what do? Probably the first thing you do before you unpack any of the boxes, right? What's the first one you unpack? The china collection. So what you do is you undo everything and you decondense your china collection, you unpack it and you put it back on display. Okay. You do the exact same thing with your chromosomes. So when you get ready to move these, you have to actually condense this down. And this is the story of that condensation. This is what you do with your chromosomes as you're condensing them. So this is chromosome condensation. It's one of the main activities of the cell cycle. So as you get ready to move your DNA, this is your 30 nanometer fiber, your solenoid. So you can see how it's just basically your nucleosomal loops, just kind of looped around in this kind of like little coil. Now, in order to condense this down even further, what you do is you basically make it into a hairpin structure. You take a little bit of a scaffold protein and you kind of cap the tips of one of these 30 nanometer fibers. And so you kind of create this little hairpin structure. Now, once you've created a bunch of hairpins from these 30 nanometer fibers, is you attach them to a long scaffold protein that looks like a cylinder, and you basically attach them into essentially like a rosette, just one hairpin at a time, plugging them into and attaching them to that central rod-like protein, creating a rosette. Now, if I were to turn this rosette structure on its side, so here is my long rod-like shaft protein, and I'm just going to look at the edge of my rosette. Then my rosette will look something like that, right? Just looking purely right at the edge of the rosette. But as I pack each rosette in behind each other, what I start to do is I start to build this sort of stacking system of DNA. Some of the rosettes are bigger than others. Some of them are smaller than others. So it starts to look like that. So if you take a look at this, this basically will look something like that, kind of like the arm of a chromosome, a cheese puff chromosome. So the cheese puff is basically a bunch of rosettes all basically packed together in there. And this is basically a mitotic chromosome. This is one that's ready to move. Of course, since you don't want to grab onto DNA, notice the other feature of the mitotic chromosome that you have is these two guys right here. These are called kinetochores. Those are like little grabbing areas. It's like a little handle to grab onto. It's a little protein pad that assembles at the centromere, which is the constriction point of a chromosome. And this is a place for those microtubules to grab onto. So you don't just grab onto DNA, you grab onto the kinetochore and you kind of grab by that handle. So never once do you grab onto DNA to move it, because that would be a disaster. We already know a thing or two about karyotypes, right? Because that's what you guys did when you did the chromosome squash. That was a karyotype. So when you're arranging all of those into, the two chromosomes on each and see which one you have, that was a karyotype. So it's your graphical representation of the number of chromosomes that you have. It's also the easiest way to illustrate what we mean by the term diploid versus haploid. So a diploid state is a situation where you have two of each chromosome. Di to ploidy, which means chromosome. And typically we abbreviate it as 2N, where N is equal to the number of chromosomes. Of course, we have 46 of them. Now the haploid set is where you only have one set of chromosomes. It also can be defined as the number of unique chromosomes in the genome. So how many unique chromosomes do we have in the genome? 23, 22 autosomes, and then the sex chromosome, right? So that's our 23. How do we get up to 46? We got two of each, right? So the haploid then is going to be the N. It's also the number of chromosomes in the genome. So you have 23 chromosomes in the human genome, in your genome. You have two of each, which makes you a diploid. Right? Now, the reason why that's important is because this isn't specific to humans. The idea of ploidy is actually something that cuts across all organisms. It's a major genetic thing. So when we learn about new organisms, we learn about their life cycle. We always talk about how much time do they spend in the diploid state versus the haploid state. Right. So there's different associations with that. We'll talk more about that later. But when you're a diploid and you have 2 of each chromosome, then those 2 chromosomes are what's referred to as homologs to each other. So they're similar, but different. Okay, what does that mean? Right? It's like, isn't that like oxymoronic? It's like either it's similar or it's different. You can't be similar and different at the same time. Sure you can. What does that mean? So they're similar in the sense that they have the same genes. They're different in the sense that they have different forms. Right, so the 2 homologs, the 2 homologous pairs would be mom versus dad. So like for chromosome 1, you would have a mom 1 and you would have a dad 1. Well, let's say that dad had black hair and mom had like really blonde hair, like bright blonde, like towhead or something like that, right? So what that basically means is both mom and dad on their chromosome 1 are carrying the gene for hair color. Same, because they're homologous. But they're carrying different forms. Dad is carrying dark, mom is carrying light. Now you have 1 of each. So that's how homologous chromosomes work. And we have to deal with that, especially in meiosis. We have to talk about homologous chromosomes a lot. In mitosis, it's not as big of a deal because all we have to do is just duplicate everything. That's all we have to do. So it's a little bit easier. But before we get ready to separate, we have to duplicate, right? So generally speaking, when we get ready to go through DNA replication, we have a single DNA molecule. So like, for instance, we'll have mom 1 will look something like this. Here's a centromere right there in the middle. It's like a little constriction point, like a little waist, like a little narrowing area in a chromosome. And then we'll have dad 1. Same thing, right? So that's before replication. Then after we go through replication, what's going to happen is we're going to make a copy of each of these, but they're going to be attached to the same centromere. So now all of a sudden, there's my centromere right there. So now this is mom 1. And now this is dad 1. So each one of these is going to be a duplicate copy of a chromosome. And they're both connected at the centromere. So each one of these is what's referred to as a sister chromatid. So for instance, this right here and this right here, these are sister, what we refer to as sister chromatids. They're identical copies of each other that are on one of the homologs. And these here are also sister chromatids. So now at this point, instead of having 2 copies of chromosome 1, you now have 4 copies of chromosome 1, 2 moms and 2 dads. So what have you done? You've gone from 46 up to, after DNA replication, you've gone up to 92, right? Which is exactly what you want to do. Because remember, you're going to be separating these apart into 2 identical piles of 46. So if you separate these 92 apart from each other in just the right pattern, then you'll drop back down to 46 and you'll have achieved your goal of creating 2 identical daughters with 46 chromosomes. Okay. That's kind of what it looks like. So let's say this is dad 1. Let's say this is mom 1. So here you can see your little kinetochore on there. And then this is like before replications. So once you replicate that, you're going to create your 2 sisters. So here's your 2 sister chromatids, right? So this one is 1, 2, And here's two sisters over here, 3 and 4. So you have 4 different chromosomes. The sisters are held together now at the centromere by a protein called cohesin. This holds your sisters together until you're ready to separate them. Okay, that way they don't just go floating off in space. Okay, you're gonna keep track of them, hang on to them until you're ready to separate them. So now you've duplicated. So that's the first half of the mantra. You've successfully duplicated, but now what about separation? So we'll get there in just a second. So eukaryotes obviously have a lot to do with replication. I don't want to get too much into replication, honestly, because replication has its own chapter. We're going to dive deep into what's going on with replication. But basically what happens is you're duplicating the genome, and then you're going to be segregating it or separating it, right? Duplicate, then separate. That's the mantra. Duplicate, then separate, in that order. And then wrapped around this duplicate and separate, we've already talked about a couple of major activities in the cell cycle, but we can actually organize and categorize these things according to the life cycle of the cell, which is what we refer to as the cell cycle. So the best way to understand what's happening isn't just to sort of try to understand what's happening with DNA replication on its own and then try to have some hope of understanding what's going on in mitosis and that sort of stuff just all randomly. The best way to understand this is to treat mitosis, a cell in mitosis, like it's one of us. Like it has its own life cycle, its own cellular life cycle. And that life cycle is what we want to basically sketch out. So everything that the cell is doing makes sense. And then since it's a cycle, it just basically connects in this ongoing loop. So let's start off then with the first phase of the cell cycle, which is what's called G1, right? So this is gap 1. For a cell, this is your growth phase. This is like your cellular childhood. You were just born, and now all you're doing is growing. You're just getting bigger and bigger and bigger. And just like us, right, with our childhood, we're growing and we're getting bigger and bigger and bigger. And so that takes some time, right? And so that's probably the longest of all the phases. But some point when we grow, we come to what we refer to as the restriction point. This is a point of decision. And we come to a restriction point as well in our life. As a cell, you have to answer a question. Do I divide? And the answer is either yes or no. There is no maybe. There is no deferring the answer. To not answer is to have it answered for you. And so it's just like us, right? So what's our restriction point as we grow older and as our body gets big enough? Yeah, do we reproduce? Do we have kids? And if you have kids, then you know that that's a yes or no question. There's not a maybe in there, right? It is either you do or you don't. And not only that, but there's a sense of commitment to that, isn't there? Like as a parent, like as soon as you have a kid, regardless of what happens, you always identify as a parent. Even a parent who's lost their kids always identifies themselves as a parent. I'm a parent. I just lost my son, right? So my son's not with me anymore, but I'm still a parent. It is like a part of your identity that like lasts with you throughout the rest of your life. And the only time that it gets changed or modified or removed is maybe if you become a grandparent. And then you sort of like promote yourself, right? From being a parent to being a grandparent. But it's a sense of identity that you commit to. Even if you're like, yeah, no, I don't want this kid anymore. I'm just going to go put them out somewhere on a street and just walk away. Well, guess what? Sorry, it doesn't really work that way. You're still a parent. You're a pretty terrible one, right? You're pretty much a deadbeat, but yeah, no, you're still a parent. So that one that you walk through that door and there's no unwalking through that door. That sounds very serious, right? But that's the restriction point that we come to in our life, and we take it very seriously, right? Cells do the same thing. And not only that, but that sense of commitment that we have as parents is also the similar sort of a thing that cells have for the cell cycle. So if a cell says yes, then it commits to the rest of the cell cycle. And if it says no, and there's many of us who said, no, me and kids, like oil and water, not going to happen, right? I'd rather spontaneously combust than have kids. That's fine, right? I mean, it's an answer, right? But in this case, this is just kind of do your job. Just kind of do what you do, right? So this drops into a functional state that we refer to as G0. So for instance, like if you're a liver cell, the reason I pick a liver cell is because liver cells have a partial regenerative capacity, so they can divide. But a liver cell who's gone through G1, they get to this restriction point, they're like looking around, they're asking, well, wait a minute, do I need to divide? Do we need more liver cells? Or are we okay? Well, if the signal that you get is like, oh yeah, you're okay, we don't need to divide, then what do? You just go do what a liver cell does, right? Do your metabolism thing, do what liver cells do, and you're good. Right. Just like for us, we're like, yeah, no, I don't want any kids. I don't want to have kids. I don't want to be around kids. That's fine. Just go do what you do. Go do whatever career you want. Go live your life however you want to live it. Be a productive citizen of society in whatever capacity you want. And that's fine. There's a lot of people who do that. Right. The last thing you want to do is be in the no category and find yourself slipping into the yes category. Right. Right, this is those situations, those oopsie pregnancies, right? Where it's like, oh, crap, I didn't really want kids, but now I kind of have one. So that's one you want to avoid. So you want to answer. This is like one of those things where either you answer the question, or the question answers you. And it's much, much better if you answer the question, right? Than having the question answer you. But let's go ahead and imagine that the cell says yes. Yes to the cell cycle. Let's go ahead and divide. What's next? Well, remember our mantra. Duplicate, then separate. So the next step is DNA synthesis, the S phase. And this is what's happening in this case is going to be the replication of DNA. Now, we got an entire chapter of what's going on in the replication of DNA. So we'll talk more about the events of what's going on in S phase when we get to chapter 14. But here you're duplicating your DNA. Now, in the next phase is what's called gap 2 or G2. Now, what's happening here? This is another pause. And so these gaps will be like pauses in your cell cycle. So here what's happening is your organelles are replicating, right? Because now you have to, it's not just duplication of your DNA, it's duplication of everything. So you have to duplicate your mitochondria, you have to duplicate your Golgi, you have to duplicate your reticulum, all those organelles, you have to duplicate them. So that's what's happening in gap 2. You also have to organize your microtubules for moving your DNA, right? So you have to start organizing all that stuff. You also do some other things here. You'll do DNA repair here as well. So if you made some mistakes during replication, this is where you're starting to clean them up and fix them. But you try to get everything just right, as good as possible before you move to the next phase, which is mitosis. Now, together, these three are called interphase. And actually, we'll be taking, like if you recall, like in the mitosis lab, where we said they had two different interphase cells, right? The G1 and the G2, right? So that's basically what we're talking about here, the pre or post replication. So you probably remember that from before, maybe before break. Depends on how good break was, right? But G2 is like a pause in the cell cycle, and you're getting everything together. You're making everything is just right before you start committing and moving yourself to mitosis, which is the division of the nucleus and the DNA. And there's 5 different subphases to this, and we'll talk pretty much in detail about those. And then you finish this off with cytokinesis, which is the separation of the cytoplasm and the cell. into two new daughter cells, which then start their gap one cycle. So a lot of times students will ask the question, well, wait a minute, how come you have this gap? You know, why do they call it gap? Where did that come from? I mean, what doesn't make any sense? Because there's obviously stuff going on in there. I mean, what's the gap thing about? This is an unfortunate hangover from research, because the way we did this was we'd watch these cells going through mitosis. And if you think about how slow mitosis is, you can imagine how tedious that was, literally just looking at a microscope like all day long and seeing cells just kind of like doing their thing. Well, every now and then what happened is cells would look like they're doing something, like in mitosis, like they're going through the anaphase thing. It's like, oh, hey, look, that's doing something. And then it would stop. It would look like it's doing nothing. And so we call that a gap or a pause in the time frame. And we didn't know what was happening eventually. We just said it's just sort of doing nothing. Right? It's later that we figured out it was actually doing stuff there. And then it would go through DNA replication. We like, oh, hey, look, it looks like it's making twice as much DNA because it's darker now. And then it's doing something, and then it would stop. It would just kind of sit there and do nothing. And we call that gap 2. Okay, so it's a second pause in the system. And then it would go through mitosis, so you kind of see it all line up and separate out. And that's where those terms come from. But when you're talking about cell cycle, you're talking about interphase, mitosis, and cytokinesis. And when you're talking about mitotic cycle, you're talking about the 5 subphases. That's probably the most common confusion that students get. Actually, they say it's like, oh, well, let's talk about cell cycle, and they start talking about things like anaphase and prophase and metaphase. It's like, no. I didn't say let's talk about mitosis. I said let's talk about cell cycle. And I want to talk about G1 and S and G2 and M and cytokinesis. So it's always very important to kind of keep clear on where discussions are when it comes to that. So here's a good question, because you guys did this, right, with your little differential cell count, right? You did kind of like a little bit of an estimation of how much time was being spent in the different phases of mitosis from the last lab. You might recall that. So the common question that comes up, how long does this take anyway? Well, the answer is not satisfactory. It's basically, it depends, right? And it depends not just on the organism, but it also depends on the cell within an organism. So here's a good example. If you take fruit fly, like an embryo, which is actively going through mitosis because it's building the little fruit fly body. Actually, it's building a little larval body. You're talking about a mitotic event every 8 minutes. So you're going through your cell cycle every 8 minutes. That's pretty quick. For a mammal cell, talking about something more like 24 hours. For a liver cell, it can be more than a year. This is the reason why liver cell regeneration is not your strategy for replacing your crushed cirrhotic liver from all of your drinking habits, right? This is the reason why we have to still do organ replacement, right? Organ donation is because this replacement strategy for cells does not move fast enough to replace all the trashed, scarred tissue from a cirrhotic liver. So here you can see that even just within the body, like for instance, skin cells, which are going through mitosis, are basically replacing themselves every like 14 days or so. So that gives you an example of 2 different cell populations in our own body. Highly moving skin cells constantly replacing themselves versus a liver cell, which is hanging out for more than a year in its cell cycle, trying to determine what it wants to do. So ultimately then, when you take a look at the sort of the actual, within the actual cycle itself, the question is, where's most of the time spent? Well, like we said, most of it's going to be in the growth phases, right? So G1 and G2. So you're paused there for a reason. You're taking your time, making sure you get it all right. The fastest one is the M phase. Think about it. Here's the reason why. In the M phase, the one thing you're doing as a mammal is you're breaking down your nuclear envelope, aren't you? That is dangerous. So breaking down the nuclear envelope is like a cellular pants drop. in public. Think about it. If you have the need to drop your pants in public, how quickly are those pants going to be down by your ankles? Or is it going to just be down there and stay there? Just walk around? You'll get arrested, right? But no, if you're in public and you have to do that, how quickly is that going to go? Let's imagine you have to go to the bathroom. You have no choice. It's either that or you're wetting yourself, right? So you drop the pants. Are you just going to linger there, grab a newspaper, read a little bit? How quickly is that process going to go? That's probably be the fastest you've ever moved in your life, because there's people all around you watching you, and there's no way you can get out of it. So leading up to that, you take your time, don't you? So if I have to drop my pants in front of people, let me get around some people that I trust. So this person doesn't look too bad. This guy over here looks like a pedophile. I'm gonna come over here, right? So you kind of take your time sort of scoping it all out. That's like the growth phases. It's like G1 and G2. You're taking your time, getting yourself prepared. But when the moment comes, you have to drop your pants in front of everybody, it's fast, it's lightning, because you're vulnerable, right? And that's exactly the reason why the M phase is fast. Okay, so we'll talk a little bit about, we're at our witching hour now, so we'll kind of just keep on going with cell cycle. We'll talk a little bit about cell cycle control. We'll talk about the events of mitosis. So we'll kind of pull it all together, and then we'll probably end up finishing, my guess is chapter 10 on Monday. So